Hello, everyone, and welcome. Thank you for joining us for today's session, the fourth in our new webinar series titled Environmental Monitoring Considerations for Gene Drive Modified Mosquitoes. My name is Justin Overcash, and I'm with the Foundation for the National Institutes of Health, where I work as part of the Gene Convene Global Collaborative. Our mission is to support and advance best practices and informed decision making for the development of genetic biocontrol technologies to improve public health. To do this, we work to identify and address key questions, provide technical advice, and strengthen capacity and information sharing. An important part of the testing pathway for new genetic vector control tools will be post release monitoring for safety and efficacy. This webinar series will highlight current vector and biodiversity monitoring efforts across Africa and introduce potential methodologies and tools that could be used to support environmental safety monitoring following release of gene drive modified mosquitoes being developed for malaria control. Sessions will take place weekly through the first week of June. Before we get started, we have housekeeping reminders. Please email events@fnih.org if you run into any technical issues. Your video and audio will be off during the webinar. The presentation will run between 45 and 60 minutes. Feel free to submit questions anytime in the chat. Questions will be addressed at the conclusion of the presentation. All webinar recordings will be posted to the Gene Convene Virtual Institute and the Gene Convene Virtual Institute YouTube channel. Today's webinar is titled Malaria Vector Surveillance for Native and Invasive Anopheles Species in Kenya, and we are honored to be joined by Dr. Damaris Matoke Muhia and Dr. Eric Achomo. Dr. Damaris Matoke Muhia is a distinguished molecular biology research scientist and currently serves as the Deputy Director of the Biotechnology Research Program at the Kenya Medical Research Institute, also known as KEMRI. She holds a PhD in molecular medicine, a master's in biotechnology, a bachelor's in biochemistry, and has a certificate of advanced healthcare management program from Strathmore University. With over 15 years of field and laboratory experience, Dr. Matoke's work has focused primarily on vector-borne diseases, particularly malaria and leishmaniasis. In addition to her scientific work, she is deeply committed to mentorship, supervising both PhD and master students, and is especially passionate about supporting and empowering young women in STEM. Dr. Matoke is also a prominent advocate for increasing the participation of African women in the fight against malaria and other vector-borne diseases. She is an active member of both global and national technical working groups, and her contributions have been recognized internationally. Notably, she is a recipient of the Aspen New Voices Fellowship in Global Health and the Kofi Annan Global Health Leadership Fellowship, and lastly, the Bill and Melinda Gates Foundation's Heroes in the Field Award. Dr. Eric Achomo is a seasoned medical entomologist and public health researcher with over 20 years of experience addressing vector-borne diseases across East Africa. He is also based at the Kenyan Medical Research Institute, where he is pioneering research in malaria vector control, particularly in the areas of spatial repellents and insecticide resistance, where he has a significant impact on global health policy and intervention strategies. Dr. Achomo leads major collaborative research efforts with international organizations, including the World Health Organization and the US CDC, with a focus on developing and evaluating innovative vector control tools in regions with high disease burden. He is also a strong advocate for strengthening local research capacity and is deeply engaged in mentoring the next generation of scientists. He holds a PhD in medical entomology and has been widely recognized for his contributions to tropical disease research and public health, receiving numerous grants and awards throughout his career. His work is also extensively published in peer-reviewed scientific literature. Before we get started, a quick reminder, I will be moderating a Q&A session following the presentations. So please feel free to post your questions in the Zoom chat at any time. Additionally, a reminder that today's webinar is being recorded and will be posted to the Gene Convene YouTube channel after the event. With that, I'll turn it over to our speakers. Damaris, if you could go ahead and share your slides, we can get started.
Thank you. Yeah, thanks a lot for FNIH for inviting us to give this talk. And I'll focus my talk on the topic of today, that's malaria vector surveillance for native and invasive Anopheles species in Kenya. As introduced, I'm Damaris Matoke Moher, a research scientist at the Kenya Medical Research Institute. So I'll start by just highlighting who KEMRI is briefly, and then do a deep dive on the vector surveillance that we've been working on. So KEMRI is a leading center of excellence and on human health research, and it has a vision on improved human health and quality of life through research, capacity building, innovation, and service delivery. Overall, KEMRI has centers, it was established by the Act of Parliament. It's a government parastatal, and it has key 7 mandates, but I'll highlight just 3, which will contribute towards the talk today. Our first mandate is to conduct health and biomedical and public health research for human health. And the second one being build human health research capacity. We have the third one, which looks at collaborate and partner with other local and international research bodies and institutions in carrying out human health research and capacity building. The fourth one, which our team within Malaria team has been supporting, is to advise the Ministry of Health in matters pertaining to health, research policies, and priorities. And fifth, to undertake scientific and technological innovations, as well as to be able to undertake discovery and transmission and enhancement of knowledge and stimulate intellectual life in the economic, social, and cultural, scientific, and the technological development. The sixth is to be able to innovate and be able to link our research with policymakers, the academia, the industry, and also the health products value chain. And finally, be able to do things that are necessary and desirable in carrying out the various mandates that we have. We've been in existence for the last 40 plus years. So since we were established in 1979. The Institute has branch centers that are distributed countrywide. What I'm projecting is a map of Kenya. And as you can see the numbers, the areas where I have centers are highlighted in blue. So we have a total, I think this is outdated, we have a total of 14 centers, and we have some 4 stations that are distributed countrywide. So we are strategically placed in the country to be able to address issues to do with malaria and other diseases. The Institute runs within 7 programmatic areas, which cut across from non-communicable diseases, biotechnology research. We have a program that looks at natural products research and drug development. The other one is public health and health systems. There's a component of One Health program. There's infectious and parasitic diseases where malaria falls under. And also there's a program that considers sexual, reproductive, adolescent, and child health. That's who KEMRI is. Infrastructure-wise, we have both BSL-2, BSL-3, and also we have enhanced BSL-3, which is able to tackle infections of pathogens such as the COVID and others, the level 4 pathogens such as the various arboviruses. And we have human resource capacity because we also have a graduate school that keeps training people, both short and long-term courses. The other facilities that is of essence is the sample receiving and management facility, which we have, which is able to monitor how many samples we have, samples that are coming in, where they come from, and also being able to support in preserving samples as far as in line with our policy or in line with our sample archiving and management policy. We have insectaries where we raise mosquitoes. We have insectaries for sandflies, etc. And we also have platforms where we do the various bioassays, which are in line with the monitoring insecticide resistance. But also we have the animal facilities where we have mice, the various rodents, the hamsters, etc., where they are maintained according to the World Health Standards. So coming to malaria, KEMRI works in line with the Kenya Malaria Strategy, and we recently launched the Kenya Malaria Strategy for 2023-2027. And the vision of this strategy is to have a malaria-free Kenya, and with the mission to be able to lead and coordinate efforts towards the malaria-free Kenya through inclusivity, equitable allocation of resources, and multi-sectoral actions. So this policy, the strategy has 3 key areas, strategic directions. The first one is the ability to be able to optimize coverage of the appropriate and impactful interventions. The second one is to accelerate and adopt the research generated by institutions such as KEMRI and others, the academia, and the innovations and also the emerging technologies in order to enhance the control of malaria and the elimination efforts in the country. The third being be able to have a resilience, because I think resilience is really important in the fight against malaria. Resilient, sustainable, and collaborative malaria control program through a transformative leadership and governance at all levels. So that's the Kenya, it was launched recently, and that's how the strategy is formulated, and it cuts across from epidemiological, entomological, and the social behavior change, and also chemotherapy. So as far as malaria prevalence is concerned in Kenya, from the data we have, based on the active surveillance and passive surveillance that has been done in the country, it indicates that approximately 70% of the Kenyan population is at risk of malaria. In 2023, the cases reported, we had around 5.5 million cases being reported in the year 2023, and this translates to around 105 per 1,000 population of cases in the country. Annually, average, we've had around 4.5 outpatient malaria cases, and this has been constant. Actually, it's been within the last 5 years. The figure has remained the same with a slight increase or decrease across, but in average 4.5 million outpatient cases are seen. So as far as incidence is concerned, we've had around 96 cases per 1,000, but this was in the year 2019, and it increased from in the report that was done in 2023 of having 105 per 1,000, 105 cases per 1,000 population. So I projected a map here that shows how malaria is stratified in the country. It's very colorful, but you can see that in the country, we have areas where we have high burden of malaria to places where there's no transmission at all. From the places which have been mapped to have no malaria at all is places like Nairobi, and this was projected using the modeling component in combination with active surveillance. But we also have places which have highlighted in white where we have low malaria transmission, very low malaria burden in those areas, even though there are a few cases reported. And we have low burden, the ones which are in page, is it page or creamish color? But to show the ones that are darker in red in color, that's where we are having high malaria burden. And where we are having deep maroon, that's where we have very high. There's high malaria burden, and also we have very high malaria burden. Of significance is places where we do not expect but we have seen malaria cases increasing. And to mention is that we have, in the 5 strata that we have the lake endemic, places which are parts of the Western Kenya. And we have also coastal endemic, places which are near the ocean, coastal Kenya. And we have highlands and also epidemic prone areas in the country. So maybe before I move to the next slide, I'd like to mention that the very high burden counties in the Kenya, we have around 47 counties. The very high burden counties have been documented to be Busia, Kakamega, Kisumu, Migori, Siaya, and Vihiga counties, which are most of them are in the western part of the country. So when we look at malaria incidence per 1,000 population in Kenya, this map projects what was projected, what had happened in 2019, and also projected for 2023. And it's in line showcasing that actually malaria in Kenya is expanding, or could we say it's increasing to areas which were known to be low transmission areas, but which are now becoming places like Turkana County, which are now becoming stable malaria transmission areas. And with the cases being recorded in unexpected areas, which we need to understand why this is happening. Is it because of climate change? Is it because of interventions not working? It is that we'll be highlighting as we go. So as we do surveillance for Kenya, for us, we undertake both epidemiological, entomological surveillance, and also there's a component of community engagement. I'll be speaking mostly on entomological surveillance, but we link towards what we've seen as far as cases are concerned. These images are part of the field work where we're doing both community surveillance, screening of patients who are sick screening of communities, households who have both clinical and non-clinical malaria, to just trying to assess on the prevalence of the disease, but also the vector sampling level in the various sites that we anticipate the vectors to be, but also doing community adult sampling using light traps and Procopac, but also engaging community and sensitizing them on the disease, especially areas which initially, like dry areas where we do not have malaria, so we needed to do education programs on the disease. So for vector surveillance in the country, we have a guideline. We have the Kenya Malaria Surveillance Operation Guideline, and this was put in place in the year 2020, 2024. I think it's being revised this year. And in this, we have specific parameters that we monitor, and as indicated in the table here. But as we monitor these parameters, we use specific entomological tools in managing this. There's the HLC, then we have mechanical aspiration. We have also the use of CDC light traps, and we also do larval surveys. When we use these tools, we're able to monitor the white marked X. When we use that tool, we're able to monitor a parameter indicated on the left, species composition, vector densities, biting rates, physiological status, parity rates, porozoid infection rates, human blood index, location of the biting time. We also look at proportion of resting indoors. We also look proportion that exit after blood meal. And finally, looking at proportion biting early or late, and then doing larval, presence of the larvas, absence and presence, and densities of the larva. So currently, routine surveillance, because as I indicated earlier, we have around 47 counties in the country. We have sentinel sites that we do entomological surveillance. There are around 14 counties, which are representative of the epidemiological zones of malaria. And in the counties, we're able to identify the sub-counties, which I think could be sub-districts. So county represents like a district. Then here we are able to go to the sub-counties, and in each sub-county, we pick 2 villages, and in each village we are able to roll out around 40 sampling points using the various tools. We do PSC, we also do human landing catches, and also we do light traps, but also we do larval collections during this. So this is how the sentinel sites, which we do routinely, we do this during the dry seasons. We also monitor during the long rains. We monitor also during the short rains, which is usually the period between October, December, short rains. And also we monitor we have a season which is dry, but cold, the June, July, which is like a winter period there. January, February, it's usually dry, hot period. And then around March to May is when we have the long rains period. So we use seasons which tend to be every quarter of routine vector surveillance. So from the data, from the routine surveillance, we've been able to identify species densities. This map might not be maybe very visible, and I think it needs to be updated. We've been able to indicate what species are found where and in what numbers. From this graph, we are able to indicate that arabiensis, which is in blue, is the dominant vector in the country. And down here is the list of the counties that we have 47 of them. However, I need to say that we've only done 45. There are 2 counties that we've not surveyed in the country. And here we've been able to see that arabiensis is the common vector in the countries. However, there is a country like Bomet where we are able to detect only Anopheles lesoni. And also lesoni was found in 3 counties, in 5 counties. That's Bomet, there's Kitui County, there's Makueni County, Nandi County, and I think, yeah, the 5 counties. So, Machakos County, that's where we're able to find lesoni. But lesoni. Then we have Anopheles gambiae also. It's not Anopheles gambiae SS. It's prevalent, but not as prevalent as Anopheles arabiensis. But also Anopheles funestus is very common in some counties such as Kericho, which are not malaria endemic, but that's the prevalent vector was found there. And places like Nyamira, the red bars is for Anopheles funestus. So we've been able to map out and through the active surveillance to show which vectors are prevalent and where they are found. So when we go as far as species diversity, I showcase this as densities, but when we look at the diversity per county, yes, we can see that Anopheles funestus, Anopheles gambiae, and arabiensis are the common vectors we're seeing in the country. But if you can see, and I'm sure Eric will be speaking more about this, places in northern Kenya, these places are arid and semi-arid, they're dry areas of the country. So Turkana, Marsabit County, Mandera, and Samburu, these places were, when we did surveillance, when they reported malaria cases, we were able to detect Anopheles stephensi. The red in the pie chart, the small pie chart shows the species that was found and some of the densities of it. But in those counties, such as Mandera, the vector we found mainly is Anopheles stephensi. Mandera borders the northern, the Horn of Africa, that's Ethiopia, on the eastern part of Ethiopia, southern part of Ethiopia, same as Marsabit and Turkana. So Ethiopia is up here. So we've been able to pick Anopheles stephensi in those places, but also there was Anopheles arabiensis. Eric will be speaking more about detection of those species there. However, we could say that still from this map, based on densities, Anopheles arabiensis is the common vector. But also we are seeing to Western Kenya, we are seeing a lot of Anopheles gambiae SS and part of Anopheles gambiae funestus, Anopheles funestus, sorry. Anopheles funestus we are seeing in blue. The blue represents Anopheles funestus and the green, light green represents Anopheles gambiae. We can see also Anopheles gambiae is distributed countrywide, but densities are lower than Anopheles arabiensis. So based on the active surveillance, we detected Anopheles stephensi. I won't speak much about it, but I'll highlight that the first detection was found in Marsabit County. Maybe I can go back to the map. The Marsabit County is northern part of the country up here, and that's the first place that the vector was detected, and this was published, and Eric is the lead author on that. But the vector was detected in containers and breeding water harvesting containers and dirty water areas, the animal watering pans. This is an animal watering pan, and also other disposed containers. That's where we found the vector in those areas. However, also this has been found in Turkana, in places where they have seasonal rivers, seasonal watering pans of animals. As you note that most of these parts of northern Kenya, they have pastoralists, they are communities that are nomads, they move around in search of pasture for their animals. They are communities where they are mobile. They tend to be mobile communities. Yeah. So as far as since that time when Anopheles stephensi was detected in Marsabit, we've been able to document it in many other places based on the intensified entomological surveillance. The presence of this has been confirmed in Turkana and Mandera counties, which border the countries at the Horn of Africa, but also now it's spreading down to into the country. So we are seeing it more in places such as Isiolo, Samburu, and also Elgeyo Marakwet. The vector has been, and also Wajir, the vector has been reported there. So through that, we were able to develop an Anopheles stephensi guideline, because then the vector was, we noted that the vector was spreading in country, and therefore the guideline was developed to be able to identify how do we tackle the species and what control measures can be put in place and how do we respond to it. So I will stop talking about stephensi because Eric is going to talk more about it. But as part of the surveillance that we've been doing, part of our data has showcased that we are finding a lot of outdoor population of the Anopheles, and this is across all species. And therefore, it indicates that either the intervention that we are implementing indoors, such as insecticide-treated nets, indoor residual spraying, are actually increasing pressure to the vector, and therefore the vector is now either coming indoor to feed and go outdoor. But there's data to show what's happening as far as vector behavior is concerned. But we are seeing increased vector densities outdoors. So when we look at blood feeding, because we needed to understand what are the blood feeding preferences of these vectors, as far as Anopheles SL is concerned, we could see that there is preference to feed on human, but also there is preference to feed on bovine. And this is mainly because of arabiensis. The data is combined for arabiensis and Anopheles gambiae. When we come to also stephensi, we also noted that actually there's a very high feeding on human, and there's also very high feeding on cow, the bovine. So we also detected mixed blood meals, and this was mainly in Anopheles arabiensis. So when we look at sporozoid infection rates by county, actually from the highest to the lowest, and this actually is in tandem with what we are seeing epidemiologically. We are seeing parts of Western Kenya having high levels of sporozoid infection in the vector, not in the patients or the people, but this is in the vector. As we collected the vectors, we were finding a sporozoid infection. Most of the vectors collected from Busia, which is the leading Malaria, and Siaya, which are the leading malaria transmission zones. We are finding that most of the vectors collected from there were infected with, they had this sporozoid. So based on our surveillance that we've done, we've been able to note that Anopheles coluzzii is now in the country. Maybe it's been there or is now in the country. So we know that Anopheles coluzzii is mostly restricted in parts of West and Central Africa, but now it was detected by Luna et al. Kamau et al. It was detected in the Kakuma refugee camp, which is northern part of the country, northern Kenya, and therefore it was a concern that what's the role of the vector as far as malaria transmission in the country is concerned? Because remember, Turkana is one of the areas that now we are seeing stable malaria transmission occurring, courtesy of identification of stephensi. Also, we've seen there's Anopheles coluzzii, but also there's a lot of Anopheles arabiensis in that county. So this just showcased, because this surveillance was done in around 5 counties in the country, but it showcases the diversity of the vectors that were detected, but it's only Turkana County that we detected Anopheles coluzzii. But the other vector that was dominant in those 5 counties was Anopheles arabiensis. We also had a part of Teso having Anopheles gambiae. So as far as diversity, especially now, we can ask ourselves, what's the difference between Anopheles coluzzii that is in West and Central Africa? How does it differ with the Anopheles coluzzii that we detected in Kenya? From this, we're able to see that Anopheles coluzzii detected in Kenya is closely related to the one in the inland of West Africa. That's Mali and Burkina Faso and parts of northern Cameroon. As far as insecticide resistance is concerned, we tested this we did target site resistance monitoring and also metabolic. We were able to detect that resistance exists. There's high frequencies of resistance in coluzzii, up to around 64%. So also we've been able to document that there's very high levels of insecticide resistance in Anopheles gambiae SL in the country. And this is courtesy of the study that was done recently, looking at the 5 counties, which are representative of the 5 epidemiological zones that we have. And yeah, as shown in the map here. So this paper by Ramaita et al. Documented that we are seeing resistance in the country. And in summary, from that paper, we're able to detect that, yes, arabiensis was most prevalent, apart from Busia, where we had Anopheles gambiae. But we were able to document that there is low to moderate resistance, which we observed in deltamethrin and primorphosomethrin and also alpha-symetrin, which are the common insecticides that are used. But we realized that this vector was susceptible to chlorophenaphyl and also chlorothianidin. So to summarize my talk, I'd like to indicate that for Kenya, based on the current surveillance we're doing, we are seeing that Anopheles stephensi, Anopheles gambiae cell, and Anopheles stephensi are still the Anopheles arabiensis are still the leading vectors, main vectors for malaria in the country, despite the reports of Anopheles stephensi and the coluzzii, other vectors which we have not yet pinned down as far as active transmission is concerned. In the country, there's a lot in existence of insecticide resistance towards most of the pyrethroids. And therefore, this led to the shift of the use of PBO nets in Western Kenya, and also an increase of IRS in parts of the country. Malaria in Kenya is expanding, going to zones which were known to be either low transmission or epidemic prone. So the disease is expanding and increasing. Therefore, it's important that the interventions that are implemented in the country are given to all population at risk. So we note from our studies that there's change in vector behavior, and also there's leading of residual transmission and dynamics in the species composition, whereby we are reporting emerging of new vectors such as stephensi and coluzzii. Therefore, it's important that we have enhanced and targeted entomological surveillance in the country. So I'd like to acknowledge KEMRI and the Ministry of Health, and also the community that we've been working with, and all the partners that have been part and parcel of this research. Thank you. So with this, thanks a lot. And with this, I'd like to invite Dr. Erica Chomo, who's going to speak more on the surveillance that we're doing and also be able to elaborate more on the Anopheles stephensi. Welcome, Eric.
Great. Let me know when you can see my screen.
We can see it, Eric.
Great. Great. Hello, everybody. And again, really glad to be here today. So I'll be delving deeper into the surveillance for Anopheles stephensi. And so maybe one of the discussions that I guess I'll talk about in a bit is this discussion about how important, how relevant is Anopheles stephensi and what we're trying to do to answer that question. And I'll also talk about a new grant that we have to try and answer that last question that I talked about, and how that might be potentially useful for developing platforms for surveillance for gene drives. So let me get right into it. So Damaris has talked extensively already about the initial detection that happened in two counties, simultaneously in Marsabit and Turkana counties, which was published in 2022. And at the moment, we're working on a follow-on report, which is as of August of last year, which is what I'll talk about briefly. But this is available as a preprint. The link is down here for anyone who might want to access and read it. I think it should be published pretty soon in scientific reports. But this, Damaris has already highlighted this as well. So we currently have been conducting stephensi surveillance in about 30 of the 47 counties. And so far we know of 7 that are positive for Anopheles stephensi. The one thing I'd like to highlight here is some of them, the ones that have the asterisks on the Marsabit, Turkana, and Mandera, have also coincidentally reported cases of vivax, which we never used to, which are not reported before. Now, this is one of those questions where we don't know whether it is actually because of stephensi that we're now seeing vivax, or we just did not have the improvement in data capture has now led us to be able to identify the presence of vivax. But this is something that we're hoping over time we'll be able to dissociate a lot more clearly. So we have been finding stephensi in a host of habitats. So in the north, people do a lot of animal rearing, and they have water traps like this, which are perfect for Anopheles stephensi. Actually, we find lots of stephensi in these concrete structures. And we've also found stephensi larvae in waste containers. So this is not your typical Anopheles type habitat. And so this was quite surprising to find them in waste debris like this. We found them in old car tires. So just getting the water from this old car tire, we found lots of stephensi. And then very, very similar to other native species of Anopheles, we find them in shallow waters like this one. This is an example, a recent example from Turkana, which I'm hoping will play out. But in Turkana, people preserve water in the concrete tanks, which are preferred over the plastic tanks because the concrete tanks keep the water very, very cool. This is a place that's very, very hot. And so because they have been told time and again that you need to cover your water sources to prevent mosquitoes, they attempt to cover those water sources. And so my team was out sampling and we stumbled upon this. So this is just the inside of this tank right here. And if you observe keenly, you should be able to see some swimming larvae. They're right there. So we found this tank was very, very productive enough of Anopheles stephensi mosquitoes. So there are establishments like this all over Turkana, and these are really, really rich for Anopheles stephensi at the moment. This is just another set of examples of habitats of Anopheles stephensi. So this is very typical of Anopheles gambiae, but you also find stephensi in habitats like this. There were all manner of water containers that people used to keep their water, and these were really good for stephensi. There's a bit of mining that happens up north in Marsabit, and they usually have water that's contained inside of shallow wells, and this is used for washing out the soil to get the gold. And in that, we did find Anopheles stephensi. So it's a very, very adaptive vector based on the environment that we've actually been able to document so far. We also have attempted to capture adults, and this has been quite a challenge. I mean, we have had lots and lots of man hours out there. We've tried a range of methods, aspiration indoors, aspiration outdoors. We've tried light traps, we've tried BG traps. And this is light traps indoors and light traps outdoors. And then we've also tried human landing catches. So far, we've been able to positively identify 33 adults using sequencing. And 12 of them were from the CDC light trap catches indoors, which is very surprising because I know a lot of the reports that we see are usually of Anopheles stephensi outdoors, and 11 in UV light traps outdoors. So as far as we're concerned for our site, the light traps have actually done very, very well compared to the BG Sentinel, which has only so far collected one mosquito. We haven't had a chance to do BG Pro traps, but this is in plan, and I'll talk about that in a moment. But we've also found some of them indoors, just collected using aspiration. We've had an attempt at longitudinal surveillance. So in the site in Marsabit where we collected stephensi way back, we've gone back multiple times. And what has happened is that every time we go back, we try and sample for both adults and larvae. So the first time we went out there, we only got larval samples. Then we went back in February of 23 and we got both larvae, both adults and larvae. Then we went back in May and we got only larval samples, no adults. We went back in October 23, which was nice. It was the raining, there were habitats everywhere. We found nothing, not one stephensi mosquito, and this was sampling over 10 days. We went back in February 24 and we had adults and larvae again. So we do not know whether these are we are observing some bottleneck effects that are associated with establishment of stephensi into this new habitat, or there's just something else that's going on that we can't account for. So this is something that we're intrigued about and is really a reason why we want to do longitudinal surveillance in these sites. We've also attempted to do genomic profiles for the populations that we find in Kenya, just trying to map and see how similar they are to populations that are found elsewhere. And so mostly we find haplotype 2 and haplotype 3. They're the most common across these sites. But in Turkana, we have found 2 samples that are haplotype 1, which matches to samples that have been found in northern Ethiopia, which is very, very strange for us. This haplotype 2 and 3 are also very, very similar. So when you look at, this is the areas where we found haplotype 2 and 3, and this is where we found haplotype 2 and 1. But haplotype 2 and 3 are also observed in the sites where they've collected stephensi, that's to the south of Ethiopia. And this could actually point to the fact that there might be movement southwards of this vector, which is potentially what's going on. I should also mention that there's a road that connects Hawassa to Marsabit. So it's one common route through which people transit from Ethiopia coming down, and we think that might be a major route for introduction of stephensi. We also plugged in this data into the MaxTemp models to try and figure out whether we could try and estimate where we're likely to see establishment of Anopheles stephensi. And using that model, we were actually able to see that it would potentially be mostly invade this north central part of the country, and would probably move down southwards to around the middle of the country, but it's likely to not affect Nairobi. Nairobi is county number 47, which is very, very tiny. It's tough to see here. And that is because just around here, there's the Mount Kenya. And so that place has a very, very high altitude and very, very low temperatures. And based on the models, some of the best predictors we saw for Anopheles stephensi were the seasonality of precipitation and the temperature seasonality. So they preferred higher temperatures, they preferred a lot lower precipitation rates, which would not be in these areas. And so for that reason, we think that we might not have stephensi getting established in these settings here, but we never know with climate change. So with that, I'd like to just talk a little bit about a new project that we have funded by the Wellcome Trust, and this is led by Ifakara Health Institute in Tanzania. And it's a project that's going to be happening across 5 different countries, so Tanzania, Kenya, Ethiopia, Djibouti, and Nigeria. So this project's goal is to assess the bionomics and relative contribution of Anopheles stephensi to malaria transmission compared to national native vectors. And the idea there is we want to try and understand how important really is Anopheles stephensi, how much resource, how much response should we dedicate to Anopheles stephensi. And to do that, we have 5 specific objectives. One is to characterize the biting and resting behavior, which I think is very, very important because that's what will determine what's the best way to respond to it in terms of control. Second is to describe the geographical distribution of stephensi across invaded or likely to invaded areas. 3 is to determine the population structure, relatedness, parasite infection status, and the spread of stephensi. And this will be done using genomic approaches. 4th is to identify and assess climatic, ecological, and social-demographic factors. So we have social scientists on the team that are going to be working together with our teams to try and figure out what human factors are important for the establishment of stephensi. And then lastly, we'll try and characterize Anopheles stephensi habitats, examine whether there's co-occurrence, whether there's competition, and what factors are important for the fitness of Anopheles stephensi. So these objectives are split into work packages, each one corresponding to the work package. So work package 1 is using spatial ecology and adaptive sampling model to enhance sampling for Anopheles stephensi. And so the idea here is that we want to try and ensure that we optimize the surveillance for Anopheles stephensi. So that if we say we have stephensi in an area, we know very, very authoritatively that we have stephensi, and if we don't, if we don't find it, then we don't have it. And this is supported by the University of Lancaster. And I'll just give an example of what we've used this to do before. So this is Malindi in coastal Kenya. So we had not done any surveillance in Malindi before. And what Professor Luigi Seda of Lancaster did for us was he was able to give us coordinates, to 30 different coordinates, and asked for us to conduct sampling in 4 structures around each coordinate in Malindi. What we realized was that there was a huge difference in the distribution of Anopheles mosquitoes that we found in these areas. And also we noticed that there was this river here, which is going to be very important. When we model this in terms of predicting the abundance of Anopheles funestus and Anopheles gambiae, we see this clustering effects, which means that you're likely to see Anopheles funestus in very, very clustered areas, similar to gambiae. I mean, you have a few spots that are outside of the common line, but you're likely to see this clustering effect, which you will not see with QLEX. So in terms of public response, everyone will say they're seeing mosquitoes, but ideally, the places that might be more important for mosquitoes might actually be very, very few, even within large geographic areas. And so this is what we're aiming to be able to do with stephensi. Survey very, very large areas and try and figure out where exactly are we seeing stephensi. And then, of course, using all of the other factors that we're collecting, we then be able to understand what factors are actually propagating the establishment of Anopheles stephensi in the different sites. And just to show you what has already been proposed for Kenya. So this is the sampling frame that's currently proposed for Kenya. This is as early as last week, Friday. So we have proposed 56 sampling sites, and it cuts across the country. Most of the north, cutting through the west, from the west to the east. And here, the numbers here represent the order of priority. So the idea is to try and prioritize them based on the numbering, and this should be really good for the model. At the moment, we're being very ambitious. We want to try and see whether we can achieve 2 sampling per week, which is quite something. So it's going to be level samples. And so the idea is we want to try and adopt a community sampling approach, identify people in each of these areas, equip them and have them conduct sampling every single week. And then we could monitor that on tablets just to see how data is coming in real time. And then every so often, we'd have the teams go out to the field and retrieve the samples for our processing in the lab. What we hope this will be able to do is to cover a range of urban and rural areas and also cover the range of transmission. So high transmission, medium transmission, and low transmission. And so in thinking about gene drives and thinking about monitoring for gene drives, I think that being able to adopt a method like this would actually be very, very useful for trying to identify how far out the gene drives are actually being propagated. So move on to work package 2. Work package 2 is the assessment of the biodynamics and relative contribution of Anopheles stephensi to malaria transmission. And here, the first thing we want to do is Latin square comparisons in 3 countries, Kenya, Ethiopia, and Djibouti, where we already have pretty good numbers of Anopheles stephensi. And here we'll be comparing a number of traps, which I'll mention in a short while. And once we've been able to identify the most effective trap, we'll deploy that to try and incriminate the vector, try and see whether we can find human blood-fed Anopheles stephensi that are also positive, and hopefully that, in comparison to alternative vectors, will be able to allow us create a hierarchy of what's the more important vector in what setting. And so these are some of the methods that we propose. This is an old slide now. We have actually had additional discussions and agreed on how we modify this work package. But these are some of the initial trapping methods that we proposed. This is the host decoy trap. This is the double bednet trap. This is the light trap, the BG traps, and then the human landing catches. And these are targeted at host-seeking mosquitoes. Then we also proposed the resting box, aspiration pots, and pit traps. I think all of these 3 have been dropped now, and we have a lot less that are proposed for targeting mosquitoes that are resting. And then for work package 2.2 is once we have the ideal method, we'll continue to conduct longitudinal sampling for 12 months, collecting adults, recording all potential confounders, and trying to use that data then to incriminate and figure out how important a vector stephensi is. Work package 3 will be using genomic approaches to try and understand, of course, migration relatedness and also the infection status of Anopheles stephensi. And to do this, we're working with Mara's group, Mara Lozenkak's group at Wellcome Sanger Institute, who are going to help build local capacity for cytochrome oxidase subunit 1 barcoding. We had proposed the announced platform, but they have a platform that's more specific to our needs, which will target about 40,000 specimens per country. And the idea there would be that we'd have Oxford nanopore sequencing capacity per site for amplicon sequencing, and run this on liquid handling robots just because of how many samples we hope to process. And then we'd have a section of the sample, 750 per country, sent out to shipped out to Wellcome Sanger Institute for whole genome sequencing. Fourth, as I've mentioned, we'll work with social scientists who will work alongside our longitudinal sampling teams to try and understand the human factors. So they will look at factors like trade, transportation routes, education status, agriculture, pastoralism, and other factors that might be leading to, and human behavior factors that might be important for Anopheles stephensi. And at the same time, we'll also be collecting data on climatic and ecological factors, which might be important for Anopheles stephensi. And the idea is to use this to sort of understand the data that we're able to collect in the 3 work packages that precede this work package. And lastly, the last work package is work package 5, where we want to try and understand the relative productivity of Anopheles stephensi across a range of larval habitats. And here, the aim mainly is to try and understand what sort of habitat we're seeing stephensi in and how productive it is, whether or not it co-occurs with other vectors, with other mosquitoes or not, or other species or not, and whether the occurrence of different species and the presence of different waters are more productive for Anopheles stephensi. And then next would be semi-field evaluations of the fitness of stephensi. And here we really want to try, I don't know if people are familiar with the works of Matt Thomas, who's currently at the University of Florida, but they have done a lot of work to try and identify the competence of Anopheles stephensi. But this has all been very using lab colonies in lab settings. And so our idea is to bring in field mosquitoes and try and replicate this kind of experiments to understand the fitness of Anopheles stephensi given different parameters. So I'll stop here by just acknowledging the different teams that contribute to this data, contribute to this effort. And I think I'll open up for questions now. Thank you very much.
Thank you, Eric, and thank you, Damaris. So yeah, now we have time for a few questions. I see we have a few in the chat, and I just want to remind everybody that if you have a question, feel free to throw it in the chat, and I'll get to it. So the first question we have is from Michael, and he said, "Thank you so much, Damaris. Can you talk a little bit about how routine vector surveillance is funded in Kenya, and what the vector surveillance workforce looks like? For example, who is doing the collections? Who is analyzing the samples?"
Yeah, thanks, Michael, for that question. I think maybe I'll talk about what has been done previously as far as funding is concerned. Routine surveillance has been funded by a global fund, and the funds come from the Ministry of Health. And but for ourselves, KEMRI, we've been supporting as far as sample processing, sample analysis, and generating data. So KEMRI is the one that now receives the samples after the ministry sends out teams. At county levels, what we did is that we trained the county team to be able to set traps, collect the samples, but then the samples are sent to the various facilities in KEMRI. There is a KEMRI Nairobi, KEMRI Kisumu, and KEMRI Coastal Kenya. So KEMRI, the research team, we are the ones support with processing the samples and generation of the data.
All right. We have another question from Nikolai. What could the expected impact of recent international aid cuts be on the Kenyan malaria strategy?
It's huge. And we recently did an analysis, and it goes to billions of Kenyan shillings. I think it's around, I think I have the figure. I'll get it somewhere, but it's huge. It goes from diagnostics, it goes to implementation of intervention, it goes even to community engagement and social behavior communication. I have it quantified. We have it. We've done an analysis on that, and there's a gap between what was being done, what was going on that has stopped, and there's a gap on what the strategy put in place to be done between now until 2027. So I'll provide that in figures as far as money, but it goes to millions of USDs.
Can I mention that
yeah.
one of the items that has already been affected is stephensi surveillance, which was, I think, 100% funded by PMI money. So that's no longer happening. And that is why our data stops in August last year, because the next surveillance was, actually we have data for December, and that would be the last that we were able to do, given that the funding was stopped in February.
Yeah, actually, even PMI was rolling out some interventions in some specific endemic sites, such as IRS and ITNs. So that has also stopped. Yeah. So they said.
So there's been a significant impact. That's what I'm hearing. Yes. The next question is for you, Eric, from Michael. Thanks, Eric. How much of the stephensi surveillance activity is piggybacked on existing vector surveillance activities, and how much of it is new?
Yeah. So we'd actually have almost taken over the national stephensi surveillance, just because what I've described to you is going to be quite intensive. Given the timeline. So usually the usual stephensi surveillance used to be 3 sites within a county, and then done once a quarter. And so what we're proposing now is sampling every 2 weeks. In many of the counties we have, I think, in some counties we have as many as 13 points where we're going to be doing collections. In many of them, I think one. But all of the counties that the government considers important for stephensi surveillance are covered under our current plan. So I think we will be able to, at the moment, cover that at least for the next year when this is planned to go for.
And I guess I had a similar question that kind of piggybacks off that, because it seems like, right, there's been a big uptick in the amount of surveillance based off the frequency that you're mentioning. And you also mentioned utilizing community members. So my question was, how receptive are community members to community sampling approaches, and how do you go about recruiting people? How much do you train them? Those kinds of things.
Actually, often so a lot of the work that we do, even though we're talking about this as KEMRI, a lot of this is led by the National Malaria Control Program. Now, the National Malaria Control Program we have two forms of government. So there's the national government, then there's the county government. The national government is sort of the arm that does policy and coordination of national level surveillance and response strategies. At the county level, there's also a county health management team. And so usually everything that we do goes through the national program, which then channels it to the county program. And then the county has community health promoters who are at the community level, at the village level. So those are the individuals we're envisioning using, but the approach for them, the approach to actually get to use them will be through the national program, and then to the county programs, and then on to the community health promoters. We've done this in the past, it's been very successful, and this is what we want to replicate again. Also, just given the scale that we're going to be working in, and two, the insecurity. So there's a lot of insecurity on our borders with Somalia and some of our borders with Ethiopia. And so those would actually be very risky for us to send out teams. And a lot of these places actually don't even have defined roads. So it would be impossible for us to conduct the surveillance on our own without utilizing members of the community. And that's what we plan to do.
Very cool. Let's see. The next question, I think this is for Eric from Brenda. I'm wondering what the duration of these work packages are, and where the data will be shared for others, and will the data be shared for others to access and collaborate?
Great. So we are actually making a presentation at the next meeting, the next Anopheles stephensi. What is it? I can't remember what the name of the convening is, but the one that said Irish at WHO convenes on Anopheles stephensi. There's a call that happens every quarter on Anopheles stephensi. So we're going to be making a similar presentation on this new grant. And the plan is that we will keep the updates coming in every quarter through that forum. So through the WHO provided engagement platform. I can't remember what it's called, but it's the Anopheles stephensi, I think, strategy meeting that happens every quarter. That's going to be one. Number two, we have actually mentioned that this is data that we want to have available to stakeholders as quickly as possible. So our plan is to share data every quarter also with the National Malaria Program here in Kenya. And so that would be one way that it's available. And then within the study itself, because it's a consortium, we will be having annual workshops, and the plan is to publish the proceedings of the annual workshop, just to ensure that we have all of the data going out as quickly as possible. But we've also put on a ton of PhD students, and our hope is that within the 3 years of fieldwork, they'll have ideally published a lot of this data. So the plan is to get this data out there as quickly as it comes. And we'll try and
it sounds like you have several venues to get it out there. That's fantastic. Yeah. The next question is for Damaris. Damaris, given the malaria status in Kenya, can you comment on the impact of the various interventions that have been in place over the years? So I think that's in reference to the increase or spread.
Yeah, thanks a lot for that question. I think specifically the interventions that we have are the main one recommended by WHO, which comes the use of insecticide-treated nets. We usually have what we call as routine mass distribution of nets, insecticide-treated nets. Then we have also for nets, we also have nets that are distributed to pregnant women and mothers through the clinics. So combination of mass net and routine provision of nets through the ANC, that's the antenatal clinics to pregnant women. We have had, I think Kenya reached around coverage of 70% coverage of the nets. And this, we believe, because we didn't do, this was not done standalone. We can't say just nets that brought down malaria to these significant levels. It was done in combination to other places you find there's a rollout of insecticide, indoor residual spraying. There's also places where we have larviciding being done. So in total, we could say that we've seen significant reduction of malaria in places such as the coast of Kenya, which went even below to almost 1% below. But we've seen also in other areas where we have had a lot of documented insecticide resistance, malaria going on the increase. So we could say, yes, the intervention have been extremely effective, but with the emergency of insecticide resistance to places such as Western Kenya, that's Busia, Bungoma, Kakamega, we've been able to see that despite the intervention being there, considering the mosquitoes now resistant to the interventions such as the use of nets, then we've seen an upsurge of cases. So in sense, we could say that malaria actually decline in Kenya has been significant, but since the 2021, it's been constant. It hasn't really reduced or gone up. So we could say we are scared because now if then resistance is emerging to most places, new species coming up, then we are going to either go back to where we were decades back.
Can I just add that that some of the really I mean, back in the 90s when bed nets first came in, I mean, entire mosquito populations were crushed. Like the Anopheles funestus disappeared from the areas like CIF for years. It took almost 10 years for it to bounce back. Anopheles gambiae to date, I think it's now that we're starting to report the occurrence of Anopheles gambiae again. So these interventions have actually been really impactful. And also, I'd like to mention, like in Homer Bay, when IRS was first conducted, they were at a prevalence of about 21%. That dropped to 4%. Busia has just had one round of IRS in 2024. And that one round of IRS, they haven't reported on the actual reduction in prevalence, but they had a prevalence of 40% before IRS. But what they have reported this year is that they have actually returned 70% of the commodities, of the RDTs, of the medicines, because they don't need them. So this intervention is actually really impactful. The challenge usually is the scale within which they can be applied, given the cost. Yeah. So it's really an issue of the cost prohibition for a lot of these interventions.
Right. The next question, also from Dorrington, is for you, Eric. Could you comment on how the longitudinal surveillance for stephensi compares with the other malaria vectors?
Good question. So it's funded as stephensi. Our plan is to do this for all malaria vectors. So at the moment, there is ongoing quarterly surveillance that happens once every quarter, and it happens in 3 sub-counties of the county. 10 houses for every 3 sub-counties. So it's not much, but it does tell you something about each specific sub-county. And the sub-counties are usually selected based on the increasing malaria burden. So at the very least, we have pretty good entomological data of the sites where we have high burden of malaria. What's going to be different a lot around this Anopheles stephensi sampling is that this strategy is very focused towards areas where we think there's a high burden of Anopheles stephensi. So in areas like Turkana and Marsabit, there's an over distribution of sampling sites. In areas further south where we think there's a lower risk, there's a lot fewer sites that you're going to be sampling. So it's not uniform like the national sampling that's currently done by the ministry. I hope that makes sense.
Yeah, that makes perfect sense. So we have another question from Richard. It says, hello, congratulations for all the presentations. Here's a question for Eric. Just to know that if in his study he shall try to rear Anopheles stephensi in insectary, it could help to explain any questions for specific objectives with any experiences. So are you going to be rearing Anopheles stephensi in the lab.
Yes, we are. So we actually, as I mentioned, for work package 5, we're actually setting up an insectary. So the challenge, we'd have done this a long time ago, but the reason why we couldn't do that is because we did not want accidental introduction of stephensi into Kisumu, where our laboratory is. And for that reason, we're setting up a new lab. We actually just got allocated space yesterday by Turkana County. So we're going to be setting up a new lab in Turkana County, which is where we shall be conducting rearing of Anopheles stephensi. So if you're interested in vector competence work, please feel free to reach out and come and work with us.
Very cool.
And actually the plan there, so let me just, we're going to be rearing both Anopheles stephensi and Anopheles coluzzii because we have both species in Turkana County. So we'll have a chance to look at this invasive species a little more.
Cool. All right, we have a question from Fred Rose. Great talks, Eric and Damaris. Can you speak briefly on the cross-border work you are doing with Uganda and how this enhances your ongoing surveillance work?
Excellent. So the cross-border work has just started. So we had an initial meeting in April. Was it March? Gosh, it's April. So we had an initial meeting between the National Malaria Control Program and its programs of each country and their stakeholders. So stakeholders being people in research like ourselves. Damaris and I were in attendance. And the plan there was to try and figure out a collaborative platform between Kenya and Uganda around our borders. I think this is just recognizing that it doesn't matter what sort of interventions we put into our countries, we would never get elimination in the border areas just because those are really one community. So some of the things that we wanted to achieve was to define what that border area would look like, define some sentinel sites, and define collaborative frameworks, which we were able to do very well. Actually, this was very, very well attended. We had the heads of the national programs from both countries attending. We had our director for primary healthcare, who's sort of slightly above the national program, also attending. So very, very well attended. We had all the people that needed to bless this union, so to say. So we're looking forward to it. At the moment, we're waiting for the final report to come out, and then we have some action items that are going to be coming out in the next quarter or so. So we're going to get some pretty good pieces of work happening there.
That's very cool.
Yeah, maybe I can add to that. I think also some of the things we felt that were keen is to be able to identify when we roll out the interventions, apart from mapping where they are, because we noted that at times you find people in Kenya who are Kenyans who have malaria moving to Uganda, being treated in Uganda, while then increasing maybe the data for Uganda saying that, and you find some from Uganda coming to Kenya. So what we've done is it's just trying, as we map out, try to identify who's really specific prevalence in the border areas, but more so, how can we share data? Because one of the things we realize is that when you don't have information on what is happening on the other side, or when they don't know, so we are able to create a data sharing framework by utilizing, I think ALMA is the Alliance for Leaders in Malaria initiative, something like that, which is being spearheaded by heads of states. So trying to develop a database, data sharing framework, but also develop a rollout of intervention framework so that they are done almost concurrently, so that we are able to have greater impact on that.
That's fantastic. Yeah, I think right, having the ability to quickly and easily share that data in real time would be something else.
Yeah.
In terms of
Yeah.
being
Exactly.
able to act quickly.
One of the first outputs to look out for is we agreed we're going to do a common prevalence map, Kenya and Uganda. So that should be coming out very soon. That's going to be the first deliverable that we.
Yeah.
Awesome.
Yeah.
All right. If anyone else has any other questions, feel free to throw them in the chat. But I know I have one since this is on right. This webinar series is on environmental monitoring. How easily could your guys' current vector monitoring efforts be adapted to monitor other insect species, right, in addition to Anopheles, perhaps environmental indicators?
Damaris, I'm looking at Damaris for this one.
Yeah, I think what we're doing, because initially what we've been doing is just focusing, let's say when I'm doing the mapping, I just do Anopheles specifically. But now we are developing, okay, maybe I could mention this from the vector division. It's called vector-borne diseases division. We've realized that it's important we have an integrated approach of especially surveillance of the vectors. So it's important that the database that they have developed an entomological database. There's a Kenya entomological database, and this will be able to populate not only surveillance of Anopheles, but we'll be documenting other species of mosquitoes. And going beyond that, we'll be able to document other species of disease-causing insects and vectors, such as sandflies. I'm talking about that because we realize that the places, especially those ones in the Horns of Africa, they have they have a lot of commonalities in a way that you're finding there is malaria, but also they have leishmaniasis. We are finding there are very high prevalence of arboviruses. So we are trying to see how can then we document this, so that we have a database that is able to showcase what vectors we have at these places. So yeah, maybe hope I answered the question. So working with both the malaria, we have division of malaria control, but also we have division of neglected tropical diseases and vector-borne diseases. So sharing that data across the platform.
So you're already expanding your platform in terms of the different types of vectors, and maybe environmental indicators or insects which might play an environmental role could be added on in the future. So very cool.
Yeah.
I think.
All right, go ahead.
Yeah, I think they talk around climate related is a big thing for Kenya, because as we said that malaria going to places that we didn't anticipate, places that were low transmission. So there's a question around what is the role of climate in this. So those are among us the questions that we want to investigate by now working closely with meteorological stations, but also doing what they call as satellite data collections around climatic conditions, the rainfall patterns, altitudes, precipitation, etc. And this will be able to inform. So yeah, I believe there's that linkages that you're forming, but climate issues, something that the country has taken seriously, and all the researchers.
Oh yeah, well, it has the potential to completely change vector dynamics and how everything operates. So that's fantastic. You guys are taking a look at that. I don't see any other questions in the chat, but I am going to ask one more, and then I'll go ahead and close this out. And that is, and I think funding is the obvious one, but what are the major challenges in maintaining and expanding the current surveillance network, particularly across the 30 plus counties that are currently being monitored? So besides, funding we can acknowledge, but what are the other challenges to further expanding your surveillance network?
I can go first. I mean, it's two that stand out. One is we have a serious problem with getting supplies, reagent supplies across most of the African countries. Supplies, I mean, especially molecular reagents are horribly expensive in Africa. And so that limits what number of samples we can usually process at a time. And for a lot of the national budget, usually that's not factored into the budget. So trying to figure out what samples to process through this process based on the budget that's available, based on the cost that we get. And also, this is also bound by inflation. So many times those prices change like rapidly. From the time I do a proposal budget and the time I get funding and actually as a project, I'm probably going to be paying double for the costs of supplies as what was there when we started. The second major challenge is the infrastructure. So a lot of the areas that we want to try and cover are pretty remote, and especially now that we're starting to do surveillance up north. Up north is mostly remote. Mostly there'd just be one major highway that goes all the way to the border. nothing on the sides. And that is why a lot of our colonists, actually for a long time, one of the first critiques we received when we presented our Anopheles stephensi data set that had the dots was, but this is all along the road. Is it that you guys are just sampling along the road? How about you get out? Like, it's not really easy to get out there. So the infrastructure is a big challenge. We are constantly running like four-wheel drive vehicles, and the wear and tear on them is horrible. The fuel demands are crazy. So the infrastructural challenges are quite something. And then lastly, of course, the labor costs that are required. We need a lot of people to be able to manage these surveillance efforts. And that's usually something else that I mean, we underestimate just how much it costs to have all of these really experienced people running surveillance. Those three for now.
Maybe just to add, some of the challenges could be now, I think because you already said resources out, but they were specific activities or interventions that were dependent on specific partners and PMI to start with. So you find that then now it's going to be a challenge to take up what the partners were doing, USAID related funded projects, such as the interventions we mentioned previously, vaccine related. But we also note that some of the areas that we are seeing malaria expanding to, accessibility, it might be a challenge. Like we had an outbreak in part of Marsabit County, but in the interior. So you find that the access road network is very poor. And that comes to adding to the cost of now trying to reach that population. That's it's likely to be a constant challenge or a country challenge, but you find that accessibility of these communities becomes a problem. But the other is rollout of interventions. It's a challenge because then if we are working with pastoralist communities, we need to think outside the box or without the box, because then the interventions that we roll out, like bed nests, don't work for such communities. It's going to be a challenge because then malaria has reached them. So it's important that the interventions that they get are customized and work for those communities who don't have permanent shelters, who are mobile communities, who now then are now the places we are getting stable malaria transmission.
Very well said. And those are certainly challenges. A lot of work to do. With that, I don't see, let me check the chat one last time. I don't see any more. So I will go ahead and close this out. First, I want to say thank you both to Eric and Damaris. Those were fantastic presentations, and you guys have obviously been very busy with all the data that you showed us. So I want to thank you for joining us today, and I want to thank everyone who joined for joining, taking a watch, and contributing to the discussion. We will hope you will join us again next week. We're going back to our regular Wednesday schedule at 11 a.m. Eastern for our 5th session in the series, where we will be joined by Dr. Mara Lonigzak, who was mentioned in this series today, who will be presenting on large-scale assessment of insects using targeted sequence approaches, opportunities for gene drive monitoring. Once again, we invite you to visit the Refresh Gene Convene Virtual Institute, where you'll find new resources, tools, and publications. While you're there, please consider subscribing to our free newsletter to stay informed about upcoming events and activities. Thank you again, and we look forward to seeing you next week.
Thank you.
Thank you.
Thanks a lot. Bye.
Bye.